Okay, so welcome everybody. And let's see, anybody have any questions about previous material? Okay. Since you guys aren't asking questions, I decided to make the quiz questions harder today. Just for fun. All right. So no questions today? That's good, because we have a boatload of material today. This is possibly one of the more complicated, if not one of the most complicated discussions we'll have, in part because there's just such tremendous activity going on in this field. It's daunting how much activity is going on in the field of cancer genetics, and hopefully today I can give you an overview and a framework that allows you to kind of understand the field and understand how the field is being leveraged for personalized cancer treatment. Again, this is kind of a dry run set of classes, and you guys' input has been super helpful. The more the better. We're contemplating inserting another class. We've kind of been talking about the fact that we don't really give adequate coverage to pharmacogenetics. We cover it a little bit during genetic testing, but we're still trying to see if we're going to expand out with an additional class. So right now we're slated to go through July 13th. If we tack on another class through July 20th. So we'll know by next week if that's the plan. So today we're going to go over cancer. We're going to talk, our case today is going to be about breast cancer, hereditary breast cancer, and for the less clinical people on the call, we're going to digress a little bit before we talk about the genetics of cancer and just kind of recap what cancer is. Then once we have a little bit of an understanding about what cancer is, that'll set the stage for talking about the genetics of cancer and how mutations lead to cancer. And then we'll finish off with some precision cancer treatment and how a particular individual's tumor profile is more and more allowing for individualized treatment. And just a reminder, the topic 12, we're going to have the ethics debate, and I keep seeing all of these ethics articles pop up all over the place, and so hopefully we're going to have some interesting discussion and some interesting points and counterpoints when we get to topic 12. So let's get started. Again, any other questions before we get started? All right. So our case today is a case of hereditary breast cancer due to the BRCA1 gene being mutated. And this is a pedigree of a 27-year-old woman who was referred to the cancer genetics clinic by her gynecologist after she was diagnosed with breast cancer. This is the patient right here. The little plus sign here means she was tested for the BRCA mutation and it was found to be positive. The negative sign in this pedigree means this person was tested for the mutation and found to be negative, and all of these other people simply weren't tested. So we know what their medical conditions were, but we don't know whether or not they were positive or negative for the BRCA mutation. She went to the cancer genetics clinic because she was concerned about her children's risk for developing cancer and about her risk for developing ovarian cancer. So her mother and two maternal aunts, so this one here and this one here, had breast cancer, and she also had a maternal grandfather, a male, that had breast cancer. Her mother also had ovarian cancer and lung cancer, and in fact died from lung cancer at the age of 52. The genetic counselor explained that the family history of breast cancer was indicative of some type of germline inherited predisposition, and patient was tested for the BRCA1 and the BRCA2 mutations and was found to have a BRCA1 mutation. And the patient asked whether or not her kids should be tested, her daughters, 6 and 7-year-old daughters should be tested. And the counselor explained that because the mutations posed little risk in childhood, the decision to have genetic testing was better left until the children were mature enough to decide on their own. So they did not pursue testing on the children, but there were a number of relatives that did want to have testing. And you can see here that there's another maternal aunt that is positive for the BRCA mutation. There's another maternal aunt over here that was found to be positive for the BRCA mutation. And because the risk of breast cancer is so high, this patient elected to have a prophylactic bilateral mastectomy. And here's a patient that had breast cancer and then underwent bilateral salpingo oophorectomy. But you can see this pedigree looks like a fairly typical autosomal dominant type pedigree, which is the type of pedigree you see in BRCA1 and BRCA2 related familial breast cancer. And when you have these mutations, when you have a mutation in BRCA1 or you have mutations in BRCA2, it dramatically increases your risk of developing not just breast cancer, but other cancers as well. And it's not just in females, it's also in males. So you can see here that women that carry the BRCA1 mutation are at least 10 times more likely to have breast cancer, and their increased risk of ovarian cancer goes way up as well. Likewise, the risk of prostate cancer and breast cancer goes up in men that have the BRCA mutation. So what exactly is this gene, and how do mutations in this gene confer a risk of breast cancer? The BRCA1 gene and also the BRCA2 gene, their gene products play a role in DNA repair. When there's double-stranded breaks in DNA, the BRCA1, the protein product of the BRCA1 gene gets phosphorylated. It gets activated by these two other enzymes, ATM and CHEK2. Mutations in these enzymes, in the genes coding for these enzymes, can also result in cancers, including breast cancer. The activated BRCA1 forms a complex with the BARD1 and the RAD51 and BRCA2, and as you might expect, mutations in the genes coding for these other components can also lead to cancer, and this complex of enzymes is then responsible for repairing double-stranded breaks in DNA. And double-stranded breaks occur periodically in the genome, and what happens is when the BRCA1 gene is mutated, that in and of itself doesn't cause the cancer, but what happens is you wind up getting lots of mutations across the genome, and ultimately some type of oncogene becomes mutated, and that then forms the stimulus for cancer. exactly why, now this particular gene product, the BRCA1 gene product, is expressed in all cells of the body. Exactly why this leads to breast cancer as opposed to liver cancer is not understood. A very interesting paradox about these tumor suppressor genes. We'll talk in a little bit about the difference between what are called oncogenes or suppressor genes. An interesting paradox has been recognized for these tumor suppressor genes. On the one hand, BRCA1 mutations appear dominant in pedigrees. So if you look at the family tree from the patient in question.
Excuse me, Bob. This is
Yeah.
Ron. There's somebody doing some kind of mechanical work on the line. If they could mute, it would make it easier for us to hear you. There's bounding and something. Thank you.
Okay, thank you. Okay, here we go. So the BRCA1 mutations, and this is the case for tumor suppressor genes in general, they appear dominant in pedigrees, but they are recessive in the cell. Recessive in the cell, in other words, heterozygotes do not form tumors. So how can it be that a heterozygote does not form a tumor? You need to have, and yet the mutations appear dominant in pedigrees. And the answer to this paradox is at least the hypothesis to answer this paradox is what Knudsen described as the two-hit hypothesis. So you have, you inherit a mutation in one of the genes, and so the offspring is born with a mutation in one of the alleles, but then you have the normal allele, and then something happens. The so-called second hit happens. That second hit might be epigenetic silencing of the alternate allele. It might be a somatic mutation occurs. It might be, as depicted in this picture, some chromosome or some segment loss. Why exactly this happens, how exactly this happens is not known, but what we see when we look at the chromosomes of cancer cells is that the normal allele has been lost. There's a loss of heterozygosity, and in fact, oftentimes that's what leads to the discovery that there must be a tumor suppressor gene remaining. So tumor suppressor genes have this interesting characteristic, which is that they appear dominant in pedigrees, but they're recessive in the cell. And what happens, the way that a tumor suppressor gene leads to the formation of tumor is that there's some second hit. Maybe it's just a random chance. Maybe it's caused by the mutation that was inherited. But whatever the reason, there's some second hit that wipes out the normal allele. And as a result now, the malignant transforming allele is expressed even though it's recessive. This slide is meant to point out that there's quite a number of genes that have been associated with breast cancer. The BRCA1 and BRCA2 mutations increase the risk of breast cancer quite a bit. Other genes increase the risk of breast cancer possibly just a smidge, but on the other hand are far more common. And so you'll see a whole spectrum of mutations in the number of genes that increase the risk of breast cancer. Possibly the best well-known are the BRCA1 and BRCA2 mutations, in part even because even though they're uncommon, when they are present, they increase the risk of breast cancer so much. So if we look at some of the bioinformatics aspects, I thought it might be illustrative to walk through a scenario and try to see what the state is with bioinformatics and informatics resources that are out there to see, because ultimately I think where we'd like to go with a lot of this genetics material is individualizing treatment decisions and doing a lot of this stuff dynamically at the point of care by having your clinical scenario or your clinical questions addressed just by plugging into these public knowledge sources. So here, let's walk through this case and let's look at the types of bioinformatics resources that we would need to access. And here, what I'm going to also try to do is point out some of the challenges we have today with integrating cancer data into EHRs and also integrating in the types of knowledge resources. And by the way, this article from Jeremy Warner, I also put a copy of this article on the download site. Really, it's a nice article. Jeremy is a clinical oncologist. He's an active member of the HL7 clinical genomics group. And he does a nice summary of bioinformatics portals for cancer. And he also does a very nice summary of barriers to integrating those portals with EHRs and of pulling cancer genetic data into EHRs. So we have a patient that has a positive family history, such as the one we looked at earlier, and she's found to be BRCA1 positive. And this is similar to the scenario we've looked at, for instance, with cystic fibrosis or with sickle cell disease, where this particular mutation generally will find that there is a ClinVar ID and an HGVS notation. And we'll also look in ClinVar and we can find that this particular mutation is pathogenic. And so we've looked at this a few times before, and I'm not going to dwell on the ClinVar scenario today, but you can look in ClinVar and you can look at this particular mutation. You can see that it's pathogenic. You can see exactly why people think it's pathogenic. And here it's pretty clear that the preponderance of submitters have deemed this to be pathogenic. So you probably can put a lot of faith in the assessment here that this is a pathogenic variant. Once we have the ClinVar ID and we've determined that this is pathogenic, we might want to communicate some type of code that can be placed on the patient's problem list. And in this particular case, even though ClinVar has links to, for instance, OMIM, from OMIM, there's no links to the specific SNOMED code. And so here, in order to come up with the correct SNOMED code corresponding to this particular variant, it requires a manual lookup of SNOMED. And this particular SNOMED code is for a BRCA1 gene mutation positive finding. So now we have this patient that's BRCA1 positive. Maybe on a problem list, we include BRCA1 gene mutation positive finding. And she subsequently develops breast cancer, and her tumor is profiled. And we're going to come back to this when we talk about personalized treatment. When we talk about a tumor being profiled, it actually means a number of different things, from genetic sequencing to looking at how the various genes are expressed. So profiling is actually kind of a loaded word that we'll come back to. And she's found to be HER2/neu positive. HER2/neu is actually, there's a gene, it's called ERBB2. It's a gene that codes for a growth factor receptor. Independently, somebody had discovered that there's a HER2 receptor on breast cancer cells, and independently, someone had determined that there's a new receptor on breast cancer cells, and was ultimately recognized that the HER2 receptor and the new receptor are actually the protein products of the ERBB2 gene. And so you'll see that the nomenclature is a little bit confusing, but basically a patient who is HER2/neu positive is overexpressing the ERBB2 gene product. Okay, the ERBB, now there might not necessarily be a mutation in the ERBB2 gene. It might be that there's other aberrations in that cell that's overstimulating this particular gene. So when we have a patient with breast cancer found to be HER2/neu positive, it doesn't necessarily imply that there are mutations in the HER2 gene. All it's saying is that the gene product is being overexpressed. And on the surface of the tumor cells, we're seeing more of these growth factor receptors than you would typically like to see. So it's not really a gene sequencing test. It's an assay looking for whether or not the product of the gene is amplified and appearing in excess on the surface of the cells. And the product is it's a growth factor receptor. And what happens is when you have excess growth factor receptor on the surface of these cells, these receptors can be stimulated, and that of course makes the cell grow. And so we can look for now personalized treatment options. And so you'll find that there's a growing number of targeted cancer treatment knowledge bases out there. I'm showing you this particular one, My Cancer Genome, not because it's necessarily the best, just because it just happens to be one that's out there that's good. And so let's say we want to look for personalized treatment options for patients with breast cancer that have ERBB2 or HER2/neu positive. And oh, by the way, this is the article from Jeremy, which is really, really quite good. So if we go to My Cancer Genome, we can do a search for ERBB2 amplification in breast, and it'll take us to a search engine where we can find targeted treatment. And when we do that, if my internet connection isn't too slow, we can find that for patients with breast cancer that have a HER2/neu positive, or in other words, have ERBB2 amplification, we see that these patients respond to a number of drugs. And in this particular case, these drugs are all FDA approved for treatment of HER2/neu breast cancer. So you'll find some information here about targeted therapies for patients that are HER2/neu positive. So this patient gets treated with trastuzumab, and this is a monoclonal antibody against the product of ERBB2, essentially trying to block it from being stimulated. And so this patient gets treated with trastuzumab in addition to her chemotherapy, but ultimately her cancer relapses. And now when her cancer relapses, we're going to do a different type of tumor profiling. We're going to do targeted gene sequencing. And by targeted gene sequencing, there's a growing number of genes that, when mutated, might suggest specific treatments, particularly in recurrent disease. And we'll come back to this as well when we talk about the precision medicine for cancer. So in this case, she has targeted gene sequencing, and we find that she has a mutation of this gene, PTEN gene, and we can find that gene in ClinVar as well. But there's another place we can look for information about this particular gene, and that's in a database called COSMIC, which is the catalog of somatic mutations in cancer, possibly the most comprehensive catalog of mutations in cancer. And so we can go to the COSMIC database and we can say, okay, Are there cases where we've seen a mutation in this particular gene, and we can take a look at that particular gene? We can click here to learn more about the gene, or we can click here to learn more about this particular mutation. And what we see in the COSMIC database is this mutation has been deemed pathogenic. Turns out this mutation has also been deemed pathogenic in ClinVar, although I will tell you that there are cases where the pathogenicity of a mutation in COSMIC is not the same as the pathogenicity of that same mutation in ClinVar. And that's partly because the way that pathogenicity is determined is in part based on predictive algorithms, and it's also in part based on crowdsourcing, which is when people submit information about these mutations, they're submitting their assessment about the pathogenicity of those assessments. So here we see that the identifier, the COSMIC identifier for this particular somatic mutation is here. And I point this out as well because when you look at the HL7 specs, when you look at the FIRE spec or you look at the V2 specs, when it comes to somatic mutations, you'll see that the standard allows for you to communicate the ClinVar ID or the COSMIC ID. And there will be a number of cases where somatic mutations have a COSMIC ID but don't yet have a ClinVar ID. And so this would be the identifier that would then be communicated. So we look and we find that there's a COSMIC ID. This is a somatic mutation, and it's also deemed to be pathogenic in COSMIC. And now we want to say, okay, this patient was treated with trastuzumab for her HER2-neu positive breast cancer. She relapsed and now has this particular mutation. Are there any other treatment options available? And we can go back to my cancer genome. And again, there's a number of places where you can do this. We can go back here and we can look at this particular mutation in breast cancer. And what we can find here is a link to this particular mutation in breast cancer. And my internet connection is a little slow today. But what we'll find here is if we look under clinical trials, there's actually a clinical trial. Patients with advanced HER2-neu positive breast cancer that have this particular mutation. And so while there may not be any current FDA-approved treatment for this patient at this point, it would be important to investigate whether she's a candidate for a clinical trial. And then, having said all that, we can communicate the information. We can communicate the information about the BRCA1 positive germline mutation. We can communicate information about the ERBB2 gene amplification. We can communicate information about the somatic mutation. And for today's example, we're going to be looking at the May 2017 ballot of HL7 version 2.5.1 lab result interface. But before we do that, let's just pause a minute and kind of recap what some of the challenges are. When you envision your goal is to have not just personalized care, but almost dynamically personalized care, where you can almost compute personalized treatment options in real time through access to public knowledge bases, we see that there's a couple of barriers. And by the way, Jeremy does a really nice job of summarizing some barriers in his paper as well. One barrier is that manual lookup is often required. There was no direct link, for instance, from the ClinVar ID to the SNOMED code. ClinVar links to OMIM, and OMIM sometimes will link to the correct SNOMED code. Sometimes OMIM will link to multiple SNOMED codes. In this case, OMIM simply didn't include this particular SNOMED code. So we had to do manual lookup in a number of knowledge bases, and that becomes a little bit daunting for typical clinicians because there's so many knowledge bases out there. It's a little bit tricky to try to keep all of them in mind. Another issue is that not all sites are searchable by HGVS, and HGVS is really becoming the language, at least for simple variants, it's becoming the language that serves as the key into all of these different knowledge bases. But what we saw for COSMIC, for instance, was that we couldn't search. We also saw that when we wanted to search for this particular PTEN mutation in my cancer genome, we also couldn't search by HGVS. Biomarker searching is less well standardized. So here for HER2 new positivity, we weren't actually looking for a mutation. There was nothing that could be expressed through an HGVS expression. What we were looking for was a gene amplification or gene product amplification. So I searched for ERBB2 amplification, but I also searched for HER2 new positive, or I also searched for a number of other search strings, and the different search strings would give different results. So biomarker, and this is recognized within the HL7 Clinical Genomics Committee, is that biomarker nomenclature is less well standardized. Pathogenicity information may be crowdsourced, and we'll certainly see there's certainly cases where the pathogenicity in COSMIC is not the same as the pathogenicity in ClinVar. Pathogenicity, when we get to our session on next generation sequencing, we're actually going to take a look at some of the algorithms that are used to compute pathogenicity. And what's being applied here in some of these cases are algorithms to compute whether or not a particular mutation is pathogenic, but also the different submitters, assert whether or not a particular mutation is pathogenic. There's no single all-inclusive site, and it's also a little bit difficult to know sometimes whether a site, how actively maintained it is. You might find a site that's very rich and detailed, but it's possible that it won't have been updated for the past 3 months. Or you might find a site and you'll find that it's good, but you'll go search some other site and you'll see that there's information between the two sites that kind of complement one another. So there's no single all-inclusive knowledge base for the association between cancer variants and personalized treatment options. Although maybe that sounds a little bit sobering, but it's not as though these issues are not well recognized. And in fact, we're seeing every day new public repositories being assembled or compilation of public repositories being assembled to try to address this. And the genomic communication standards are still maturing. So for instance, in this case, we have a ClinVar ID and we have a COSMIC ID. And the question is, well, which one do you communicate? And this is a somatic mutation, so do you want to send the COSMIC ID? Well, some resources won't necessarily recognize the COSMIC ID, but other resources won't necessarily recognize the ClinVar ID when it comes to somatic mutations. So there's a little bit of a fuzziness there. The other issue in some of the standards is that when we talk about communicating a germline mutation, we can talk about whether that mutation is a heterozygote or homozygous. With somatic mutations, it's a little bit more complex, and the standards really still haven't formalized the approach here. Because, for instance, when you're doing a biopsy of a tumor, and tumors are heterogeneous to begin with, there's different mutations in different parts of the tumor. And so trying to communicate some type of quantitative estimate about just how prevalent a particular somatic mutation is still a bit tricky, still not really well addressed in any of the standards, whether they're HL7V2 or FHIR. So let's now communicate the information, and let's take a look at the May 2017 LRI ballot. And really, the way this works, it's similar to the V2 message we looked at before, where you have LOINC panels. And if you plug in this particular LOINC code, for instance, you can see, you can just look at the hierarchy here in LOINC. So these are all of the LOINC codes that comprise the panels. And basically the way these work is you include, and you can see here the cardinality for the different codes. So just think of each one of these LOINC codes as a specific field. And we have the overview. So these are LOINC codes that correspond to the overall study. You know, what genes were tested? What were you looking for? Was the overall findings positive? Were the overall findings negative? And section 1 is required. And then you can have these other different times. You can have 0 to many section 2. These are discrete variants. And so for each discrete variant that's found, you can include LOINC codes from in here. And these discrete variant LOINC codes will tell you what was the HGVS for the mutation. Was it pathogenic? And what's the corresponding SNOMED code, etc. Sometimes you have complex genetic variant panels. Complex genetic variant panels are where multiple mutations correspond to a particular phenotype. And we'll come back to this in later sessions. We won't be looking at section 3 today. We'll be focusing on section 1 and 2. And we'll also be coming back to section 4 in a later session. The panel includes pharmacogenetics, where you can indicate, here's the drugs I'm interested in. And based on these genetic findings, does the lab predict that this person is going to be a rapid metabolizer, for instance, or going to be an ultra-fast metabolizer or a slow metabolizer? And as a result, what the medication usage implications are. And if we look, and by the way, for this particular message, I did a little bit more detailed example, and this is also on the download site. If you want to take a closer look at this example in a little bit more detail. I cut a lot of it out just so it would all fit here on the screen. And so again, you have report section 1, which are variables that apply to the overall study. You have report section 2, which are the discrete genetic variants. And in this particular example, there's nothing from the other report sections. And the main things to highlight here is what I'm reporting here is somatic mutations, not the germline mutations. So this is how a lab might communicate the fact that I analyzed the tumor specimen. And in the tumor specimen, now this was a targeted sequencing test that we had ordered. And so the lab says, these are the genes that we specifically tested for. These are the, these are the mutations that we specifically tested for. And our results are positive. And we found this PTEN mutation, and again, it's a somatic mutation. There's a lot of these fields in the message are optional, and that's in part because they're superfluous if you have the ClinVar ID or if you have the Cosmic ID. But I include them here just to kind of help illustrate what some of these different fields mean. What gene was studied? What was the transcript reference sequence? What was the DNA change? And you can see all of this stuff is given in the complete HGVS syntax already. What was the amino acid change? Well, in this case, the amino acid change here was from an arginine to a serine, and that's it's a missense type of mutation. meaning the amino acid was changed from one amino acid to another, so that's a missense mutation. But again, all of this stuff is optional. All you really need to send is the fact that here's the specific mutation that was seen, and that this is a somatic mutation. Okay. So let's do a little bit of a quiz. You guys ready for a little bit of a quiz? Okay.
Let's go.
What is the, go?
Let's go, yeah.
Let's go. What is the function of the BRCA1 gene product? Anybody remember?
Double-strand DNA repair.
Double-stranded DNA repair, that's right. That's right. BRCA1 is generally, not always, but generally, a germline mutation or a somatic mutation?
Germline.
Right, right. There are cases of sporadic breast cancer where you don't have a BRCA1 mutation throughout all the cells in your body. You only have the BRCA1 mutation in the tumor itself. But by far the more common scenario is you inherit the BRCA1 mutation. It's in all the cells of your body. And then you have the second hit, and then from there you wind up with your cancer. Name a bioinformatics challenge when looking for treatment information on a recurrent cancer.
Manual lookup in various data sources.
Exactly, exactly. That's great. Manual lookup is often required. And let's see here. Here's a couple of others. Not all sites are searchable by HGVS. Biomarker searching is less well standardized. Pathogenicity information is crowdsourced. There's no single all-inclusive site. So we got a few barriers. We got a few things to keep us busy for a while. Okay, so now we're going to shift to looking at what is cancer, and we'll just have a few slides on what is cancer. And I think most of you on the call probably will already know a lot of this stuff, but it's going to set the stage for the genetics. So worth going over. And obviously this picture here is probably going to be pretty obvious to all of you, where you start having the tumor cells, they proliferate, they look different, they don't look like mature cells, they look like immature cells, they then invade, and then they metastasize. And I think, early on, and, it wasn't so clear to me as it is now that, cancer is not so much yes cancer, no cancer. It's more of a progression or a transition from normal to a state exhibiting different types of malignant behavior. And some of the things you'll see with cancer is the deregulated cell proliferation. We'll be talking about deregulated cell proliferation quite a bit, because obviously these cells keep cycling and keep multiplying, and that causes a lot of the problem. We also see failure to differentiate. Also, when you get a lot of DNA damage, oftentimes that will trigger the cell to autodestruct through a process called apoptosis. But the apoptotic pathways are also affected in cancer, and so the cells can't self-destruct. They can't kill themselves. They have to keep growing even though they know that they're damaged. Genetic instability is a hallmark of cancer, and as we'll talk about in a little bit, there could be hundreds to thousands of mutations in cancer cells. And, part of the problem is some of the genes that get mutated are the genes responsible for DNA repair. And once those genes get mutated, then you have increasing amounts of genetic instability. You have loss of replicative senescence. So you probably have heard of these things called the telomeres. The tips of the chromosomes are referred to as telomeres. They don't get replicated the same way as the rest of the chromosome during the DNA replication process. And as a result, every time the chromosome is replicated, the telomeres shorten a little bit. And after about 25 to 50 replications, the telomeres have gotten so short that any further attempt to replicate that chromosome can lead to death of the cell. It can lead to apoptosis. But sometimes in a lot of cancer cells, there'll be mutations that will result in an enzyme called telomerase being active, and that will keep the telomeres long, and that will allow the cells to keep dividing way past the 25 to 50 level that normal cells have to stop at. So this loss of replicative senescence means cancer cells don't reach a point where they have to stop dividing. We see increased angiogenesis. There's factors produced by the tumor cells that stimulate blood vessels to grow into the tumor cells to nourish them. We see invasion and metastases, and then we see evasion of the immune system. And we'll come back to this. In fact, we'll talk a little bit about immune therapy. I think a lot of people may remember that Jimmy Carter had metastatic melanoma with metastases to the brain, and his tumor was put into remission using therapy that circumvented the tumor's ability to evade the immune system. So tumors have clever ways of evading the immune system. In some of those cases now, there are FDA-approved treatments that can get right at that evasive mechanism. So, sobering facts. Cancer is a result of a somatic mutation in a body cell. And it's not as though these somatic mutations in body cells are rare. In fact, when you consider that there's 10 to the 14th cells in an adult, and there's 10 to the minus 10th replication errors per base per cell division, and you estimate that there's 10 to the 15th cell divisions during a person's lifetime, you recognize that there are thousands of mutations in every cell of our body. Thousands of mutations in every cell. And most of these mutations, they don't do anything. They're in introns, or they're in the regions between genes. But every once in a while, one of those mutations just happens to be in a tumor-suppressor gene or in a tumor oncogene. So it's not as though mutations in a cell is some exceptional event. They're happening all the time, and at least from a probabilistic mechanism, you could say, well, okay, every once in a while then, one of these mutations is going to happen to be a pathogenic mutation. And if there happens just by chance to be another pathogenic mutation in that cell on top of it, now all of a sudden you begin to set the stage for cancer. So what's going on in cancer is you have these successive mutations, this stepwise progression. Okay, so you start off with a normal cell, and perhaps just by chance, you have a mutation in a key gene, and that might result in some phenotype that's not yet considered cancer. It might be considered, benign neoplasia. Then there's another mutation, then there's another mutation, and now you have a polyp in your colon, or now you have a cyst. And with progressive mutations, you now have progressive neoplasia, and now the pathologist looking at that tissue under the microscope says this is frankly malignant. So this picture here illustrates the stepwise progression of cancer. There's multiple mutations in cancer cells, hundreds to thousands. The other thing that this picture illustrates is the intra-tumor heterogeneity. And so you'll have, in this clump, so you'll have this tumor cell, and then it'll split, but one of the daughter cells will then go on to have some mutation, whereas the other daughter cell may go on to have a completely different mutation. And so one of the challenges with assessing the mutations in a tumor cell is that they're heterogeneous. And in fact, the metastases might look different from the original tumor, in part because it's thought that you need specific mutations to allow a tumor to metastasize. So if you biopsy this part of the tumor, you may get a false sense of the metastatic potential of the tumor, and it may not look the same as if you do somatic testing on the metastasis. People are trying to get around this by studying what are called liquid biopsies, where you actually just measure, you get cell-free DNA, tumor DNA floating around in the blood to give you more of a statistical mix of what's in the tumor. But this picture does, I think, nicely illustrate the intratumor heterogeneity. That's a reality when you're studying mutations in cancers. So.
And these are only, I noticed these are only carcinomas, so you're not including, it's different for sarcoma or for neuroendocrine tumors, which can metastasize?
It would be the same. I mean, this picture just happens to just show carcinomas.
Okay.
But
okay.
All of this mechanism would be the same.
All right, thank you.
So, what is a cancer? Well, what is a cell and what does a cell do? And, how does a cell live and how does a cell, spend its life, and then how does a cell ultimately die? And cells have a lot of options. They can just kind of sit there and do their thing, like a muscle cell. Some cells, like stem cells, differentiate. Some cells grow and divide, and some cells die. And when we look at these kind of, cell outcomes or cell options, these are the key options that are affected by cancer mutations. So the cancer mutations are generally mutations that factor somehow into growth and cell division, or that factor somehow into cell death. So cancer mutations interfere with cell death, or cancer mutations stimulate cell growth and cell division. Okay. So let's turn away from what is cancer to now let's start looking at cancer genes. Let's try to get a better sense of what cancer genes are and where they play a role. But before we do that, let's just talk a little bit about some of the terminology. You'll hear tumor-suppressor genes and you'll hear oncogenes. And any type of categorization of genes or any in cancer, it's good, but it's not perfect. There's no, there's a number of cancer-causing genes that don't necessarily neatly fit into a tumor suppressor bucket or an oncogene bucket. And you'll see different sources will talk about the BRCA1 gene as a tumor suppressor gene, but other sources will say, well, no, that's a DNA repair gene. And so this is kind of a useful framework to think about mutations, but it's, it's not the central dogma of cancer genetics. But you have this notion of tumor suppressor genes, and they regulate cell growth and proliferation. In some ways, you can think of the gene products of tumor suppressor genes as holding a check on the cell cycle. It holds the cell back. It keeps the cell from cycling, or it allows the cell to die. And when you interfere with tumor suppressor genes, you're in essence removing the brakes. You're now letting the cell increase its cycling. Oncogenes, on the other hand, promote cell growth and proliferation. So if you have an oncogene, it plays an active role. The oncogene is going to stimulate cell growth, whereas the tumor suppressor gene is going to inhibit cell growth. Tumor suppressor genes are recessive at the cellular level, although again, they appear dominant when you look in pedigrees, and that's because of that two-hit hypothesis. The second copy of the gene, for whatever reason, is frequently lost, and so tumor suppressor genes are recessive even though they appear dominant on pedigrees. Oncogenes, on the other hand, are dominant. You only require one copy of the gene to be mutated. Heterozygotes can manifest tumors, as opposed to tumor suppressor genes where heterozygotes cannot manifest tumors. And oncogenes gain function and tumor suppressor genes lose function. And in inherited cancer syndromes, most of the inherited cancer syndromes, so adenomatous polyposis coli or BRCA-induced breast cancer, etc., most of these germline mutations resulting in inheritable cancers are due to inherited tumor suppressor genes. There's only a couple of cases where oncogenes are known to be inherited. One example of that would be the multiple endocrine neoplasias. Some common tumor suppressor genes that you might be familiar with would be BRCA1 and BRCA2, and the adenomatous polyposis coli gene, APC gene, that results in familial adenomatous polyposis. Common oncogenes you might be familiar with would be the Philadelphia chromosome, the chromosomal abnormality that results in CML. And then also there's the RET oncogene responsible for multiple endocrine neoplasia. The other thing I should point out here when we're looking at cancer genes is if you take a cancer, you might find that it has hundreds to thousands of mutations, but only a small percent are driving the malignant transformation. And so you'll hear people talk about driver genes versus passenger genes. So for instance, when you have a BRCA1 mutation, you have problems with repairing DNA damage. And as a result, you get all kinds of mutations. You can have hundreds of mutations, you can have thousands of mutations, but only some of those mutations are pathogenic and some of them are just along for the ride. So you'll hear people talk about driver genes, you'll hear people talk about passenger genes. A tumor can have hundreds to thousands of mutations, of which only a small percent are driving the malignant transformation. And COSMIC, which is perhaps the largest catalog of somatic mutations in cancer, lists about 600 known oncogenes or tumor suppressor genes. So maybe about 600 genes that are mutated now actually drive cancer. All of the other mutations that we're seeing are at least presumed to be passenger mutations. Okay. So cancer disrupts the cell cycle. And this is a picture we looked at before, where as the cell is going through its cycle, there's a number of checkpoints along the way. And imagine, if you will, that these checkpoints get out of whack. We look, for instance, at this thing here called activated MPF. And MPF is, it stands for maturation promoting factor. It's comprised of proteins, which we'll come back to. And I'm not going to get too detailed into pathways and specific names. I'm going to delve into some of the pathways in part just because they're super fascinating. I'm also going to delve into some of the pathways so that you kind of have a better understanding of how mutations disrupt the cell cycle or interfere with apoptosis. The MPF, which is the maturation promoting factor, it's comprised of a couple of proteins called cyclin and cyclin-dependent kinase, has to be activated in order to allow for the cell to progress past this checkpoint. So the cyclin and the cyclin-dependent kinase have to be activated. Well, imagine, if you will, an oncogene that activates MPF even when the cell wasn't ready to go. You know, maybe there was some damage, or maybe not everything that was necessary in order to proceed to mitosis was ready to go. Imagine, if you will, an oncogene that activates MPF. Well, that's going to stimulate the cell to divide, even though there was DNA damage. So these checkpoints are important, and there's hundreds of genes and hundreds of enzymes that play a role in cell cycling as well as in these checkpoints. And that's why, at least when we look at cancer, we're seeing something a little bit different than we're used to seeing with a normal disease. I mean, you take sickle cell anemia, where the relationship between genotype and phenotype is nearly 100%. And now you look at cancer as the other extreme. Well, you know, two different people with breast cancer can have very different mutations. They can have different driver genes that are mutated. They can have different passenger genes that are mutated. So the correlation between genotype and phenotype is very complicated in cancer, in counter-distinction to, you know, cystic fibrosis or sickle cell anemia, where there's a much tighter genotype-phenotype correlation. And part of the reason is because there are so many proteins and so many enzymes involved in cell cycling and apoptosis. And really, imagine, if you will, it's really when enough of them get out of whack that you wind up getting into problems. Okay. Cancer is also we see disrupted apoptosis, and apoptosis is, you can think of it as normal cell death. Sometimes cells die because they're inflamed or diseased, and that would trigger an inflammatory reaction. But apoptosis would be a more orderly. The cell says, okay, I've lived my life, it's time for me to say goodbye. And the cell goes through a very orderly process where it degrades itself. The bits and pieces are gobbled up, and there's no inflammation. And sometimes, for instance, a cell will say, look, you know, I'm at a checkpoint, you know, but gosh, the DNA is so damaged, you know, I can't, in good faith, continue to allow this cell to divide. And so the cell will trigger its apoptotic pathway and commit itself to dying in an orderly fashion. So the apoptotic pathway is very important, as well as the cell cycling pathways. And this picture is a little bit overwhelming. It's really quite a pretty picture, I think. this is a picture, pathways in cancer. I got this picture from the KEGG database. And by the way, just as a reminder, all of these pictures and all of the web addresses, you can find them all in the notes for the slides. The KEGG database is manually drawn pathway maps where the researchers try to assemble experimental knowledge on metabolism and a lot of other functions and try to show the complete pathways. And so, for instance, what the researchers have done here is they try to summarize everything that's known about pathways in cancer. And you can see how a lot of these pathways converge on apoptosis. And you can see how the cell cycle has a central role in a lot of these pathways, and how a lot of these pathways, when they're out of whack, lead to proliferation. And an understanding of pathways is a big deal in cancer because sometimes, while you may see a mutation in this gene up here, understanding its pathway means you can still target an intervention downstream in that pathway. And so you'll see that these pathways are very big. But on the other hand, this is a pretty confusing picture. It's just more of a fun show and tell. If we were to try to take these pathways and summarize at a high level what types of genes are being mutated in cancer, this picture might give you a little bit of a better idea. And again, I think it's difficult, if not impossible, to really neatly categorize the genes that drive or contribute to the development of cancer. So I've tried to show you here the major classes of genes. The other thing is that this is orthogonal to the notion of oncogenes or tumor suppressor genes. So for instance, mutations in growth factor genes, some of them will act as oncogenes, whereas some of them will act as tumor suppressor genes. Mutations in genes that code for growth factor receptors, some of them will act as oncogenes and some of them will act as tumor suppressor genes, etc. So this notion of oncogene and tumor suppressor gene is orthogonal to this categorization of the types of genes where mutations lead to cancer. So growth factors can be mutated, and you can imagine, for instance, if a growth factor is mutated, it might hyperstimulate a growth factor receptor. You can imagine if a growth factor receptor is mutated, it could hyperstimulate the signal transducers, and it could trick the nucleus into thinking that that cell is being stimulated even though there's no stimulus for that receptor. When growth factor receptors bind to their ligand, what happens is they communicate that information into the nucleus by way of signal transducers. And signal transducers are oftentimes these things called protein kinases, where they activate a protein such as RAS, and then RAS in its activated form will activate some other protein, and ultimately the signal transducers will carry the fact that the growth factor receptor has bound growth factors at the cell surface and carry that information into the nucleus, where then transcription factors will activate specific genes. And those specific genes then can have a stimulatory effect on the cell cycle, or might have an inhibitory effect on apoptosis. Also, you have these DNA repair genes, and like BRCA1, and these can result in problems with checkpoint inhibition. They can result in problems with apoptosis. And so you kind of get a sense of, there's a boatload going on here in cancer with these pathways. But really, I think we can largely think of the types of genes responsible for cancer as falling into these large buckets. And just to further illustrate the fact that cancer is a result of mutations that lead to increased cell cycling and decreased apoptosis, this is just another picture that shows the growth factor binds to the receptor. The receptor then stimulates protein kinases or the signal transducers, which then stimulate transcription factors to activate particular genes and translate those into various proteins. So here, for instance, assume that a patient has inherited the APC gene, and the APC gene, the adenomatous polyposis coli gene, is a tumor suppressor gene, and that results in familial adenomatous polyposis. And the loss of this gene then begins the transformation from normal cells to malignant cells by changing the phenotype from normal to hyperproliferative. Maybe you start to get a polyp. The promoter region of an oncogene might then be hypomethylated, and thereby removing some of the inhibitory effects of and allowing that oncogene to be transcribed more. That then results in the oncogene CRAS stimulating the phenotype to develop into an intermediate adenoma. And we have these successive mutations that ultimately lead to cancer. But I will point out that this is one pathway, but this is it's not the case that all colon cancers follow this pathway. It's really more, there's hundreds of proteins, hundreds of genes involved in the cell cycle and the apoptotic process, and it seems to be more, you get enough of them, then the cell cycle gets screwed up and the apoptotic pathway gets screwed up, and your phenotype gradually transitions from normal to more and more malignant appearing. And if you get additional mutations thrown in there, then your tumor now has a propensity to metastasize. So this gives you one example of a progression of mutations, but it's not the case that all tumors would follow this same progression. So let's look at just a few examples in a little bit more detail. So this is the RET oncogene. This is the gene responsible for multiple endocrine neoplasia. And the RET oncogene codes for this protein. It's a growth factor receptor. And normally what happens is the growth factor receptor binds to some ligand, okay? And when the ligand binds to the growth factor receptor, that then phosphorylates the receptor, activates the receptor, and allows the receptor to begin phosphorylating and activating a number of signal transducing proteins. And so in the normal state, the ligand binding to the receptor then communicates downstream and ultimately sends signals into the nucleus that says, it's time to cycle. Let's let this baby grow. So let's stimulate proliferation and suppress apoptosis. And in the oncogene, in the mutated copy of this gene, what happens is this particular receptor is what's called constitutively active. It's active even without having bound its ligand. So the receptor is just sitting there, and it's stimulating the signal transducing proteins. It's suppressing apoptosis, and it's stimulating proliferation, even in the absence of a bound ligand. And that then is a cancer stimulus. If we look at the Philadelphia chromosome, now a lot of folks probably have heard of the Philadelphia chromosome before in CML, chronic myelogenous leukemia, which is the result of a translocation. A bit of chromosome 22 swaps places with a bit of chromosome 9. The ABL gene from chromosome 9 is moved next to the BCR gene on the modified chromosome 22, or the Philadelphia chromosome. These two genes are then juxtaposed, and you get this novel protein, the BCR-ABL fusion protein. The function of BCR is not known, but the function of ABL is this is a signal transducing protein kinase. So this is a signal transducer. And what happens here is, having fused to BCR, it now acts as a signal transducer, even in the absence of being activated by a growth factor receptor. So this guy now is transducing signal right and left, even without being told to do so by some type of growth factor interacting with a growth factor receptor. Okay. And this is, I think, the last example, and this picture is pretty complicated, and part of the complication is because the biology is so complicated, where you have such a cascade of inhibitory factors and excitatory factors. And, you know, Ron asked this question early on, well, how the heck do you know where it all balances out? And it's really just a dynamic equilibrium, you know, between all of the inhibitory factors and the excitatory factors, which determines, you know, today the cell is at rest and tomorrow it's going to start to grow. And it's possible that you've heard of P53 and PRB because they play such a central role in cancer. P53 is a protein. It's the protein product of the TP53 gene, and it's the most commonly mutated gene in cancer. And it plays a central role in cell cycle regulation and apoptosis. When you have P53, it stimulates another gene to produce P21, and P21 puts the brakes on cyclin and cyclin-dependent kinase. And by putting the brakes on these, it doesn't allow the PRB to be phosphorylated. And again, it's kind of a confusing picture with all these positives and negatives, but the PRB binds to a factor, but if you activate PRB, you release the factor, which can then stimulate the cell cycle. And so, as you might guess, mutations in P14, P21, P16, cyclin, CDK, a lot of these will have effects on the cell cycle. A lot of these will have effects on apoptosis. And P53 and PRB play this very central role, and that's why there's a lot of activity right now looking specifically for anti-cancer treatment that can target mutations in P53 or PRB. So let me just pause there for a minute. Actually, I'll pause before we get into our quiz questions. Let me just pause there for a minute. Any questions? That's a boatload of material. Anybody have any questions? Okay, how are you guys doing? You guys still out there?
Yeah.
Okay. So
everyone shy at me?
Everyone's shy. I know. Don't worry, you're being recorded, and if you get the answer wrong, everybody will know. So don't be shy.
I'm not shy.
Okay, so name a cellular pathway that is dysregulated in cancer. And I'm going to give you a hint. Huh?
Apoptosis?
Apoptosis, yeah. Yeah. So the cell cycle pathway, apoptosis, signal transduction pathway, all of those pathways, if they're dysregulated, can result in cancer. Which of the following is true about cancer? A given cancer always has the same mutations developing in the same order? Or a given cancer from two different people can show different mutations?
I'm waiting for somebody to say B.
Okay. So B, right? A given cancer, two people with breast cancer can have very different mutations, very different somatic mutation patterns. Okay? All right. Ready to move on? Talk a little bit about precision cancer treatment. Okay. So when you think about, well, let's go back to this picture that I love so much. When you think about this, when you think about, somebody's going to have a mutation here, 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 it stands to reason that different treatments might work different in different people. And so this notion of precision treatment means to focus on the specific molecular abnormalities in an individual patient. We might focus on the specific molecular abnormalities to look for treatment, whether that's some type of cancer therapy. We might also look at the specific molecular abnormalities in an individual patient in order to help determine eligibility for clinical trials. And of course, clinical trials are very big in cancer. And there's all of these different pathways affected. So let's take a little look at precision cancer treatment. And there's what I want to talk a little bit about is how people are assessing what type of treatment to give. And so what we're going to talk about are these things called biomarkers. We're going to talk about gene profiles, and we're going to talk about gene sequencing. These are three different ways for looking at the individual characteristics of a particular patient in order to determine the best therapy. Some of you may have seen the FDA's press release. What is this? Hot off the presses, right? Like a week ago? Right? FDA approves the first cancer treatment for any solid tumor with a specific genetic feature. They said that Keytruda, Keytruda can be used to treat cancers that illustrate microsatellite instability high or mismatch repair deficient. So it doesn't even matter what kind of cancer it is. If your tumors are demonstrating these biomarkers, then Keytruda is indicated. So let's take what exactly do we, and when we talk about biomarkers, in biomarkers, we've talked about HER2/neu. A biomarker is not necessarily a specific genetic mutation that you can assess for. A biomarker might be, how high is your cholesterol? A biomarker is some objective physiologic measurement, okay? And you may recall when we talked about genetic polymorphisms that across your genome, we have these things called short tandem repeats. We have areas in the genome where I might have 3 or 4 repeats, but at that specific spot, you might have 6 of those tandem repeats. Somebody else might have 8 of those tandem repeats. And so there's a lot of different spots, thousands of spots throughout the genome where we have these short tandem repeats. And, we look at these short tandem repeats as a way of doing DNA fingerprinting or as a way of doing paternity testing. And sometimes these short tandem repeats, when they do grow within a gene, can cause disease. But we also see that when you have mutations in the DNA mismatch repair proteins, when you have mutations in the genes that shepherd the DNA replication process, you'll start seeing all kinds of screwy things happen. And one of the things that you'll see happen when you are having problems with DNA replication is that when your DNA replicates, the parent gene may have had 4 of these tandem repeats, but in the daughter, maybe it's now 6. And so the short tandem repeats, or these microsatellites, are unstable. And the instability of these microsatellites is a sign that this particular tumor has a boatload of mutations, and it's going to be a very aggressive, very malignant acting tumor. So when we look here, for instance, if you take a normal body cell, and let's look at this one particular marker, we see that one of the alleles has this many copies of the short tandem repeat, and the other allele has this many copies. But if you look in the tumor, we see that, wait a minute, oh, there's all kinds, there's multiple copies, there's multiple lengths of this particular short tandem repeat. Marker 2, we don't see a problem. But marker 3, we also see that there's a lot of variability now in the number of short tandem repeats. And we call this microsatellite instability, and it's a sign of mismatch repair. It's a sign that there is a tremendous amount of mutations going on in this particular cancer cell because the enzymes responsible for repairing the cell have gone awry through mutations. And when we see that with a cell like that, these cells often learn how to evade the immune system. Okay, they can evade the immune system. When T cells try to kill tumor cells by recognizing them as having foreign proteins on their surface, the PD-1 receptor, if it finds that the cell is expressing the PD-L1 ligand, the PD-L1 ligand tells the T cell, hey, wait a minute, it's checkpoint, I'm a friend, don't destroy me. And so this class of drugs, such as Keytruda, which are called checkpoint inhibitors, and Keytruda was the drug that was given to Carter for his metastatic melanoma. They block the PD-1 receptor, or other checkpoint inhibitors block the PD-L1 ligand. They then don't allow the tumor cell to trick the T cell into thinking that it's a friend. They don't allow the tumor cell to evade the immune destruction. And so with Keytruda, now the T cell can go in and destroy the tumor cells. So Keytruda doesn't specifically act on the microsatellite instability, but rather microsatellite instability is a marker for an aggressive tumor, just as HER2/neu is a marker. Sometimes we'll see overexpression of PD-1. That can also be an indication, another biomarker for Keytruda. The other types of biomarkers, which we're going to come back to when we talk about genetic engineering, are foreign cell surface proteins. So there's other types of immunotherapies that I think we'll better talk about when we talk about after we've introduced genetic engineering. Anyhow, so biomarkers can lead to precision cancer treatment. This other thing called gene profiles can lead to cancer treatment. And this is, I think, interesting in that here what's going on in gene profile is we're looking at 50 genes. And we're not looking for mutations in this particular panel of 50 genes, but rather we're looking at the extent to which the RNA, the messenger RNA, is expressed in the cell. What you can do is you can take a cancer and you can extract the messenger RNA, and you can compare it with normal levels of messenger RNA, and you can see that for certain genes in green, the messenger RNA for these particular genes are being overexpressed, whereas you can see here in red, the particular messenger RNA for those genes is being underexpressed. And this also, to me, speaks of big data analytics in these large cancer databases. When you get to the point where your clinical decision making is based on, no longer 3 or 4 or 5 different tests, but now 50 tests plus some physiologic variables, and you have to balance the results of these 50 or 60 tests in order to come up with treatments. This is the direction I think we're going with precision cancer treatment and big data analytics. Here, what we'll see is that for breast cancer, when you look at the gene profiling for these particular 50 genes, studies have shown that they can be lumped into four different profiles. And these profiles correlate with survival. So, for instance, one signature might have this green survival curve, and another signature might have this red survival curve. So if you take a patient and you look at their particular gene profile, and you possibly also combine, physiologic variables, you can now get a sense of, oh, your tumor looks more like this tumor profile or this tumor profile. And from there, we can get a much more quantitative sense of your prognosis, and possibly even use that information to say, you have a good prognosis, we're not going to give you this treatment, or you have a poor prognosis, we're going to treat you more aggressively. So the gene profiling in breast cancer, for instance, is being used to, for instance, determine the duration of adjuvant endocrine therapy. And other types of gene profiles are being used to determine prognosis, which may then lead to a determination of the type of chemotherapy or the duration of chemotherapy. So again, we're not looking specifically at gene mutations. We're looking for the extent to which these genes are expressed, overexpressed, or underexpressed. The other way that precision cancer treatment is being approached is through gene sequencing. And there's a growing number of cases where knowing the specific mutation will provide an indication for a specific treatment. This is a small list from an article in 2015. There's a knowledge base out there that's much more current, but just to give you an idea, now you can do targeted gene sequencing, and there's products out there where you can say, okay, I want to do targeted gene sequencing and it will look for all of the mutations for which there are FDA-approved treatments, or for which there's clinical trials, or et cetera. And so gene sequencing is now being looked at, and in many cases there are FDA-approved treatments based on the results. And imatinib, for instance, was one of the first small molecule treatments specifically targeting genetic mutation, the Philadelphia chromosome mutation in CML. And that's it. That's it for today. That's it for today. We got through that fast. Any comments?
No, I'm just looking at, in the laboratory to see, how things are reported for next generation sequencing. I'm looking at this Mayo Clinic, Mayo Medical Laboratories, OncoGene for hematologic neoplasms. And one thing I like about their website is that they attach sample abnormal reports, so you can see really what do they report. So so they do report the lists of defects and things like that. But then there's a very neat there's an interpretation that will talk about therapeutic implications, disease associations, mutation effect, et cetera. So I think maybe a lot of that is algorithmically even derived, but certainly reviewed by medical directors, and because it's not as if you do this test and you can get, like a pregnancy test, you could get 3 results plus negative and indeterminate. Here you're looking at a huge number of genes by NGS. So
yeah, and this will be something important, I think, for us to talk about in the session on next generation sequencing, because depending on the technique, you could literally find thousands of mutations, many of which are of unclear clinical significance. And so oftentimes what the labs are doing today is they're reporting back what they decide to be the significant ones, right? And 2 different labs might report back a different set of variants. And it might be the lab fails to report back a variant that was significant. And there's lawsuits over this stuff. And I think the trend, well, maybe it's just a wish, maybe it's not a trend, maybe it's a wish, is for the results of the next generation sequencing to be available. Now, the lab might want to selectively say, here's what we think are the important variants, but I think where we want to go is that the providers should have access to the complete set of sequencing information, and the patients should have access to the complete set of sequencing information, so that they can poke around, or so that a year from now, as new knowledge comes to light, you can go back to that particular tumor genome or the germline genome, say, oh, now that we know a lot more, let's take another look. Right, and
that's really true, because I remember reading that Steve Jobs paid, when he had his neuroendocrine tumor, his pancreatic cancer due to his PNETs, there was really considered kind of a rare disease because there is not as much research as there is in standard cancers, and he paid $100,000 to have a total genomic profile done of his tumor. But what do you compare that to? That's one man's genomic
Yeah.
That's one man's tumor, you know. I mean, how do you correlate that? How do you make sense of that? In the absence of research and in the absence of having many people who have, with certain kinds of cancer, where you know that, you know, you can correlate conditions and prognosis because you have the clinical information related to those genetic profiles.
Yeah, that's a really important point. Great.
So much, let me see, what is it? So much information, so little knowledge.
Yeah, these databases, boy, they're just popping up everywhere. These huge compilations, you know, where they're trying to get, like for cancer, you know, all of the variants, all of the phenotypes, trying to do correlations. And, you know, I think the bioinformatics aspects and the clinical informatics aspects are, you know, kind of sort of coming together. A lot of people are sharing the same vision for how all of this needs to come together to more directly impact the patient at the point of care. Okay. Any other comments? Anything good, bad, different about the presentation, about the material? Suggestions? Okay. Alrighty, guys. Okay. Thank you, everybody. Have a great week.